Only half of you are here so far. And I do appreciate you being on time. That's good. That's good. Okay, the live stream is up. So for the sake of live stream, good morning, good morning, good morning. Get your macromolecule chart out, and we're going to be looking at lipids. So if you go to your Canvas model, we're going to start with Canvas today. So we're going to fill things in on the chart from Canvas today. So go to Canvas, go to lipids, have your chart handy, and I'm going to pull my chart up. All right, they're coming, they're coming. Oh, I already have that up. What do I need? I don't need that. All right, good morning, good morning. If you just came in, we're pulling up our macromolecule chart and we're going to Canvas and we're looking at lipids. I'll pull my share screen up in just a second. Good morning, Andre. All right. I think I covered nobody in this class. Was the other class? Everything is graded if there, and everything has, if you need, if you have something missing and you need me to extend the availability date, because I think on most of the assignments, the availability date has closed. So in order for you to access a late assignment, you're gonna have to let me know so I can change the date. I usually try to have all the assignments um, open, and tonight's parent open house, so this is what I'm going to tell them as well, is that I try to make everything available for at least a week past the due date. Okay, so that gives you time. The due date, after the due date is late, but it's still available for you to access it so that you can at least get a late grade. Um, but after that, if you try to do it, you won't be able to. And I want you to let me know that you need to get access to it if you were sick or for some reason weren't able to get it in on time. Um, under normal circumstances, I'm not going to change the late date, but I will and Canvas will grade it late accordingly, depending on how many days late it is. Canvas automatically takes points off for being late, but a grade is better than a zero, always. So you want to make sure that you turn in all of your assignments. Like I said, right now, I think you all are wonderful people, and I don't think you have anything outstanding for me at the moment. So, all right, all of my slow moving computer. I've got too many windows open. When you got too many windows open, everything moves slow. Now I'm going to jump back and forth, so try to keep up with me. I'm going to jump back and forth between your macromolecule chart and what we're taking from Canvas. Now, I have added a few things to my chart that you didn't have on there before, but for the most part, you are going to have more things on your chart than I have on mine. I have the basics, so the bare minimum, but I want you to have everything. Okay. And also keep in mind that when we finish this unit, you're going to turn in your macromolecule chart for a grade. Okay. So hopefully as we go through this, you are keeping up with of the, oh my goodness. Every time I share it just totally alters my Zoom view. Okay. All right. So this is what we should have so far. All right, we have done proteins and we have done carbohydrates. Now, like I said, you should have more on in your boxes than I have. Every once in a while, I do add something to it, but for the most part, you have more writing in your first two columns than I do. Okay, because you have taken them from Canvas and from the videos that we've watched and placed that information on the box. You want to have this so that when the test comes, you're not running all over the place because we have 5 macromolecules. So the test is going to be on 5 macromolecules when we finish the unit. And you don't want to be all over the place looking for answers. You want to have that one chart in front of you that's going to help you answer all the questions. Okay, so this is for you. I'm going to grade it, but this is for you. This is to help you to organize everything in one spot. Also, remember that you have an end of year test, and at the end of the year, this will also help you with this particular topic of macromolecules. So, um, brief Backing up briefly, macromolecules are large molecules necessary for living things. In themselves, by themselves, they are not a living thing, but they are things that we need in order for us to survive, or living things need to survive. And again, macro means that they are big molecules, but still microscopic to you, but they're still bigger than an atom. So when we looked at the organization of life, you started off with atoms, and atoms make molecules, and then molecules make larger things like tissues. Okay, cells and tissues, that kind of thing. So we're looking at macromolecules. We looked at proteins. You should have in a function in your box. Now, what we're going to look at too, so I'm going to put this in all the boxes, but the fact that all of these molecules are made up of some type of backbone of carbon. So the proteins are made up of carbon, hydrogen, oxygen, and nitrogen. And in particular, the amine group always has a nitrogen attached to it. Carbohydrates only are made up of carbon, hydrogen, and oxygen. Okay, and then that brings us to our lipids today, which are made mostly of carbon and hydrogen. We have some oxygen and some phosphorus. So you don't have this in your box, so please add that to your box. Okay, and then we're going to go to Canvas. Some of this I got from Canvas, but while I have the picture up, copy anything that you don't already have for lipids. So you don't have made with carbon, hydrogen, with some oxygen and I'm just going to add words to it. Okay. Anybody need me to copy these pictures into the chat? Great. So far, so good. Because I remember we spent a lot of time with this last time. All right. So you're good. You're good. You're good. All right. So, and then, like I said, when we get to Canvas, we're going to go over these some more. But while we're here, let's look at putting lipids are insoluble. Insoluble means that they do not dissolve in water and they store high amounts of energy. Okay. All righty. The molecule above it, so remember this first, it's distinguishing molecules. And we're going to look at this when we get to the notes, that the molecule has, it's a funny shape to it. It has a top and a bottom, or like a heads and tails. And the head part is hydrophilic, which means um, it likes water. Hydrophobic, and anytime you have a phobia, you have a fear of or you repel. You have a phobic end, which is the tail end, which does not like water. Okay. Hydrophilic is attracted to water or allows water, and hydrophobic is resistant to water. All right, so then you should have your monomer. A monomer of fat is called a fatty acid. Okay, and the fatty acid, let me make this a little bigger for a minute. Fatty acid, oop, won't get big. Why won't it stretch? I know we just won't go beyond the, and one of the reasons I did my box like this is so I can give myself more room. And then you can do the same thing. You can make your lines bigger so that you can stretch your picture bigger. The picture won't get any bigger than the column, but when you add words, it will make the bottom column move out the way so that if you have to add things as you will, um, it'll move. But you can adjust these columns to be whatever size you need them to be. All right, so our monomer is called a fatty acid. And notice the fatty acid has a head and a tail, so to speak. Okay. Now, when we go over to the polymer, we're going to take that one fatty acid and we're kind of turning it sideways. And now there's three of them. So so this one picture that I have going down for the fatty acid. Now we want to turn it, and now there's three of them, and their polymers are. And I think you all had the picture, but you didn't have the title. So please put the title triglyceride on your chart. Let me make that a little bolder on mine. So this molecule is called a triglyceride. Here we go. Okay, and that's the polymer. All right, and examples there of different types of lipids are fats, and sometimes we use the word fat and lipids interchangeably, but fats, waxes, oils, and steroids. All right. Now we're going to come back to the chart because I'm going to have you put something to Canvas. So we'll be back and forth between Canvas and the chart. Everybody with me? All right. Oops. Okay. All right, let's start at the beginning. My page just keeps jumping on. Again, a lot of times we use that word interchangeably. And then down here at the bottom, you have all the examples that we just put on the chart. So if you didn't get those before I switched my page, there they are. The examples are fats, waxes, oils, steroids. Okay, those are examples. And when we put this other part under the other column, in that they are insoluble and they have high amounts of energy. Now, I mentioned on your chart, you should have carbohydrates function is to provide you with energy. And we talked about the fact that a lot of times when people want high energy, they carbo load. Because when they talk about carbo load, they're eating foods high in carbohydrates because that's the job of a carbohydrate, is to give you energy. Okay. If you don't, if you want to lose weight and you decide to take the carbohydrates out, the body still needs energy. So what the body will do now is it'll go to the lipids. It'll say, I need energy. Lipids have energy. I'll start breaking down the fat, which is what diets are designed to do. They start deciding that I'm going to make your body go someplace else to get its energy, and it breaks down the lipids next. Now, if you deprive it of that, it'll go back and start, well, I still need energy. I'll start breaking down the proteins. So this is when people get really, really thin, because it's the next thing after, if it has no fat to break down and it has no carbohydrates to get the energy from, it's going to get the energy from the proteins. It's going to get it from somewhere. Okay. So when those three are gone, then you're near death, because there's no place else to get energy from. Okay. All right. So we're going to look at lipids. So in your first column, we put this already. So make sure that you have this. So I put it in initials, but make sure that you understand that C is for carbon, H is for hydrogen, and O is for oxygen, and P is for phosphorus. Okay. So they are the macromolecules made up of those elements. And we have our examples on our chart already. All right, characteristics. Here again, I think we put this on. So that's the same thing you put on the chart just now. This you did not put on the chart. So this part, I do want you to add to your chart. So I'm going to stay right here, and you flip to your chart, and you add under function that they act as waterproofing. They act as chemical messengers, such as steroids. So steroids, what a steroid is a chemical messenger. They store energy, and they form cell membranes. So you want to put this under function. So I'll give you a few minutes to do that. All right, again, the first box you already have, so you shouldn't be writing that one, because we already did that in our second column. But in your first column under function, you need to write 1, 2, 3, 4. So again, your line will move as necessary. I don't have it on my chart because it wouldn't let me copy and paste. All right, somebody came off mic and I'm echoing. All right. And again, please use this time that I give you to keep up, because later on, this chart is for you. Later on, when it's due, you don't want to say, I didn't get this. What'd you put for that? I'm telling you what to put on the chart now. This is going under the function column, the importance of lipids. And yes, you need all four. All right. So when we think of waxing, you're kind of waterproof. Okay. When water kind of just is this not going to soak into your skin. If you walk out in the rain, the water is just going to roll off of you. Ducks and plants, the top of a plant is a waxy surface, so the water just kind of rolls off of that. Birds, their feathers have kind of a waxy um, coating to them so that they can fly around in the rain and not get soaked. Again, the chemical messengers that we're talking about, not all fats are chemical messengers. Steroids are chemical messengers. The fats that we store are not the all of the things that we take in. And you want to watch a video that said that one of the reasons, there are some fats that we need to have, and we need to eat them because we need them to build these materials for our bodies. We need them for cell membranes. We need them for insulation. We need them to make steroids. So there are certain fats and oils that we need to have. All right. All right. And this is in Canvas. So if you need to go back, you know where to find it. Okay. Triglycerides. We already wrote that word down. That's our polymer. They are used because, and let me go back to that for just a second. Look at how big this molecule is compared, and it may not look big because the picture is small, but imagine, compare this, see how many hydrogens and carbons are linked one behind the other. So these can be tremendously big. These can be long chains of carbon and hydrogen, carbon and hydrogen. And as you see, there's only a little bit of oxygen up at the top, but it's mostly carbon and hydrogen linked together all the way down. These are huge. And then when you study chemistry, and actually when we talk about, we're going to do a little chemistry later on when we talk about respiration. All those little lines represent a bond between one atom and another. In order to break that bond, it releases energy. So compared to breaking all these little bonds to get energy, and you're looking at the sugar and they got one little line connecting one to the other, there's more energy in a fat molecule than there is in a carbohydrate molecule. Okay, so look at the pictures so that you see the difference there in structure. And again, just remember that when we digest food, we have to break the chemical bonds between these molecules, and that's how we release our energy. So more energy is being released from a triglyceride than there is from a polysaccharide. Okay, back over here. Okay. So there's a lot of energy located in the carbon-hydrogen bonds of a fatty acid attached to the glycerol. The glycerol is that backbone. When broken down, this energy can be used by the body, or the molecules can be synthesized for storing energy for a later use. In other words, you're going to take these molecules and store them as another kind of molecule that the body can use. Everything that we eat is not in the form that we can use it. So the body breaks it down, takes it apart, and reassembles it into something it can use. All right. If you listen to TV at all when they're talking about selling diet things that are good for you, but the argument is always saturated fats versus unsaturated fats. And I'm going to let the guy in the video talk more about that, but you do need to know the difference between those. All right. These are our examples. So again, you don't have to write this down, but you do need to know what they are. So steroids are the signaling molecules or hormones, and cholesterol is a steroid. You all heard about cholesterol. All the commercials are talking about watch your cholesterol. Cholesterol is good, but cholesterol can also clog your arteries and create too much problems. So too much of a good thing is never a good thing anymore. So you have to watch your cholesterol levels. Wax is important in animals, but mostly in plants, and they provide waterproofing. Okay, so steroids and waxes are examples of lipids. All right, we're going to watch these two short videos. I'm going to stop talking.
Welcome to Biomolecules,
This
the
one is
lipids.
low, so you might want to turn
Lipids
your
are organic
volume up.
molecules that are insoluble in water. In biology, lipids' main functions include storing energy, signaling, and acting as structural components for cell membranes. Let's examine three types of lipids: neutral fats, phospholipids, and cholesterol. Neutral fats, also called triglycerides, are three fatty acid chains attached to a single glycerol molecule by dehydration synthesis. The fatty acid chains are the building blocks. While these chains can differ in length, each one has an even number of carbon molecules. Neutral fat stores energy fuel, insulates body tissues, and cushions and protects organs. Saturated fatty acids have single bonds between the carbon atoms. They originate from animal sources and are solid at room temperature. Other fatty acids don't bind their maximum number of hydrogens due to double bonding between carbon atoms in the chain. Fatty acids with one double bond are monounsaturated, while those with two or more double bonds are polyunsaturated fatty acids. Unsaturated fats originate from plants and are liquid at room temperature. Phospholipids are modified triglycerides, where a phosphate group replaces one of the fatty acid chains. Phospholipids, or phosphoglycerides, have a nonpolar fatty acid chain portion and a phosphate polar portion. Two layers of phospholipids are the chief component of all cell membranes. In a cell membrane, the two layers self-assemble so that their water-soluble heads, or hydrophilic heads, form the surface and interior of the membrane, and the water-insoluble tails, or hydrophobic tails, face each other. Cholesterol is an organic molecule known as a sterol, or modified steroid. It helps build and maintain membranes and composes over 30% of all animal cell membranes. The cholesterol molecule has 4 interconnected carbon rings. Cholesterol helps to stabilize all animal cell membranes. The body uses it to synthesize steroid hormones, including sex hormones, and those of the adrenal cortex. It's also used during vitamin D and biosynthesis. Congratulations! You've completed biomolecules, the lipids.
All right. So we'll look at cell membranes later when we talk about cells in particular. So you're going to have to, that same picture that they just showed you, the cell membrane, you're going to have to know all the parts and all the movements going in and out of a cell. So you'll see that again. And we'll talk about the importance of the way that the bilipid layer works. All right. Let me see. I'm gonna slow him down because he talks way too fast. All right, I think that's good. Now this one big video on macromolecules. What they've done in Canvas is they've chunked it up into pieces. So he's going to review carbohydrates a little bit, and then he's going to move into lipids. And they're going to stop him just before he gets to the next one.
Our own use. All for me. Now we as human beings can't grow fruits or tubers, so we have to store our energy in a couple of different ways. The way that we tend to store carbohydrate energy is in glycogen, which is very similar to amylose or starch, but has more branches and is more complicated. It's basically made up of the glucose that we have left over after we eat, and it sits in our muscles really ready to use, and it's also stored in our livers. It's generally a pretty short-term store. If we don't eat for like a day, pretty much all of our glycogen gets depleted. But over the longer term, the way that we store our energy is through fat. All of our mom's worst enemies, the fat, which turn out to be actually really important and are the most familiar sort of a very important biological molecule, the lipid. Lipids are smaller and simpler than complex carbohydrates, and they're grouped together because they share an inability to dissolve in water. This is because their chemical bonds are mostly nonpolar. And since water, as we learned in the previous episode, despises nonpolar molecules, the two do not mix. It's like oil and water. In fact, it's exactly like oil and water. And if you've ever read a nutrition label or seen this thing called the television, you're probably pretty conversant in the way that we classify fats. But then, 99% of us have no idea what those classifications actually mean. Fats are made up mainly of two chemical ingredients, glycerol, which is a kind of alcohol, and fatty acids, which are long carbon-hydrogen chains that end in a carboxyl group. When you get 3 fatty acid molecules together and connect them to a glycerol, that's a triglyceride. These feature prominently in things like butter and peanut butter and oils and the white parts of meat. These triglycerides can either be saturated or unsaturated, and I know that when we put the word fat and saturated into the same sentence, it sounds like an evening at KFC, but here we're talking about being saturated with hydrogen. As you hopefully remember from our first lesson, carbon is very nimble in how it uses its 4 electrons. It can form single or double or even sometimes triple bonds. This means that if the carbon atoms in a fatty acid are connected to each other with single bonds, all of the carbon atoms end up connected to at least 2 hydrogen atoms, and one of them picks up a third. So the fatty acid is saturated with hydrogen. But when some of the carbon atoms are connected to each other with double bonds, all of those carbon's electrons are spoken for, and so they're not able to pick up those hydrogen atoms. This means that they're not saturated with hydrogen, and they are unsaturated fatty acids. To demonstrate, may I direct your attention to this jar of peanut butter. Here you can kind of see both kinds of fat. The liquid stuff you see at the top here, that is the unsaturated fat, which we generally think of as oils. The pasty stuff down here also contains lots of unsaturated fat, but also contains saturated fat, which doesn't have any double bonds, so it can pack more tightly and form solids at room temperature. And there are also other fat classifications that you've heard of. Trans fats, which everyone tells you never to eat. They're right, don't eat them. They don't exist in nature and are basically unsaturated fatty acids that instead of kinking go straight across, and so they're extra super bad for you. Don't eat them. Omega-3 fats are fatty acids that are unsaturated at the 3 position, which is like right there. Then that's the only difference. But the reason why these are important is because we can't synthesize them ourselves. They're essential fatty acids, meaning that we need to eat them in order to get them. All this is starting to make me pretty hungry. But before we get to more food stuff, there are some unappetizing sort of lipids that we also need to talk about. So remember that triglycerides are 3 fatty acids connected to a glycerol. Swap one of those fatty acids out for a phosphate group and you have a phospholipid. And these make up cell membrane walls. Since that phosphate group gives that end a polarity, it's attracted to water. And the other end is nonpolar and it avoids water. So if you were to scatter a bunch of phospholipids into some water, they would hydrophilic ends sticking out to face the water. Every cell in your body uses this natural structure to form its cell wall in order to keep the bad stuff out and the good stuff in. Another class of lipids is the steroids. Steroids have a backbone of 4 interconnected carbon rings, which can be used to form hundreds of variations. The most fundamental of them is cholesterol, which binds with phospholipids to help form cell walls. But these can also be activated to turn into different lipid hormones. And so now we approach the most complicated.
All right, so he's going to switch to the next one. So when we do nucleic acid, you'll see him again and he'll pick up from where he left off. All right, so that's kind of a review of what we already have on our chart. Take a minute while I do attendance to work on this activity. So do the matching, practice, and check your answers, and then make sure you can answer these questions down there. So I'm going to stop sharing for a minute so I can do attendance. Then I'll be right back. While I'm doing that, you do the matching. Tomorrow we're going to do the lipid Nearpod, and if well, we're going to do nucleic acids now, but we won't have time to do both tomorrow. But we're going to do at least lipids tomorrow. So practice this. Read that. If you finish before I get back on to share, go ahead and look at nucleic acids. All right, that is the next thing on our chart. So I'm gonna stop sharing to attendance. You look at that, read that, make sure you have everything on your chart that you should have. And if it stops sharing, I could do attendance. Right. So if you finish reading lipids, move on to nucleic acids, because that's where I will be when I switch back. Okay. All right. Remember, I do attendance by looking at GoGuardian. So when I look at GoGuardian, I should see you looking at lipids, not looking at me, which is what I'm seeing. All right. Everybody should be looking at lipids. And if you finish and doing the practice vocabulary section. So that's what you need to be doing. The practice vocabulary section. Then we're going to move on to nucleic acids. Just a reminder that tutoring is on Tuesdays and Fridays at 1:35 to 2:05. So if you need a little extra help, please let me know. And I'll be available to you at that time. Okay, so I think everybody is here. Yes, they are. So that's a quick attendance. One of the things he mentioned in the video about fats is that, like I said, we take those molecules apart and build things that we do need. Okay, I'm looking in the chat and I don't understand what conversation between Imani and what does peanut oil mean?
It's the stuff
Right, well,
in the video, you know, the peanut oil that goes at the top of the peanut butter.
oh yeah, have you ever seen your jar of peanut butter do that? No, that happens. You let it sit long. Well, you almost get your peanut butter and eat it just before it does it. But if you let peanut butter sit for a long period of time, the unsaturated the oil will rise to the top. So you get this nice layer of oil, and it's not bad. You can just mix it back up with the rest of the peanut butter. Just so know that it is part of, but it will separate and sit at the top. And then, like he said, that's the saturated and unsaturated part of it, just separating out. But if that happens, it's not bad. Just mix it back up. But I guess when you get a jar of peanut butter, it don't last long enough for it to separate at your house. So that might not be something that, yeah, yeah. And hopefully you didn't throw it out. Well, check the expiration date for sure. But if it's not old, um, you don't have to throw it out. You can just mix it. Yes, you could have. Yeah. So you see, you learned something today. Learned something that helpful. All righty. So next time you see that oil on top of the peanut butter, you won't throw it out. But yeah, yeah. So you don't have to, like I said, do check the expiration date because you don't want it to, because oil gets rancid. But for the most part, if you let it sit for a little while, the oil will separate out of it. So just mix it back up. All righty. But he was talking about omega-3s, and you hear a lot about omega-3s, and you need to, some people take omega-3 vitamins because that's called an essential fatty acid. You need that, and the body doesn't make that, but it needs it. So that is one type of fat that we have to have. So you need to eat the kinds of meats, fish, salmon that contains those because the body needs to have them. All right, we're going to move. That being said, anybody else got something to say, to share? All right, so we're going to look at nucleic acids now. So we've got our three macromolecules. We're on macromolecule number 4. And we're almost, because it's only 5. Water's not going to take us long because you're real familiar with water. That's probably the one you're most familiar with. All right, so if you slide on to the next Canvas slide, you should be at nucleic acids. Okay. And we're going to spend a lot of time talking about this later on when we do genetics. We have a whole unit on genetics. But again, we're just interested in the fact that it's a macromolecule right now. This is just an intro. This is not really telling you anything. It's just, now they are different. So that's the one thing that is going to, we're not going to eat all the other ones we got from our food. We're not going to eat nucleic acids. This is going to be something that is going to be manufactured in our cells. And I'm
Created using Powtoon.
okay. All righty. You have, let's look at the macromolecule chart for a minute. And let's make sure we have these pictures, and then we'll add some stuff to the chart after that. Come on, get bigger. I'm trying to zoom and it won't zoom. My screen is frozen. But again, we have a picture of a nucleotide, which is 1 unit on a DNA molecule. And then the whole DNA, again, we're going to spend time talking about this. So we're going to talk about the fact that DNA is double-stranded. It looks like a spiral staircase. So we're going to take all these nucleotides and link them together to make this spiral staircase called DNA. And my thing is still is not. Does anybody need these pictures? Everybody have the nucleic, it's a nucleotide, and we don't call it so much a polymer, but the whole double-stranded DNA and RNA molecules are what we refer to as polymers. But anytime we link the monomers in together, that's a polymer. Okay, I don't know why. Okay, there it just blinks. Yay, finally. Okay, there we go. All right, does anybody need these two pictures? I can put them in the chat so that you can have them on your, you need, okay. Well, we'll do water later. Today I'm only worried about um, so I'll put like the last time I had to go back to. All right, picture number 1. Oops. Okay, uh-oh. I made my picture disappear. Okay, hold on a second. Let me see if I can make it go back. Go back, undo, 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 undo. Where's the undo? Where's my undo? I might have to put this picture back. Yep, I will. I can't find my undo button. All right, let me do this one. Don't mess that one up. And it did the same thing with that one. Ah! Oh, I know what I'm doing wrong. Okay, let me get those pictures back. And yeah, yeah. All right, so we want, okay, let me get the pictures back. And I'll look them up. And then this way, when I copy it, I'll just put it in the chat and then I'll put it on my chart. But my computer is taking a nap. I tell you what, while I'm trying to deal with my tech issues, y'all read the Canvas page for nucleic acids. And then when I finish getting all these things back in place, we'll talk. Okay. DNA. There we go. That's the picture I had. Copy image. All right, that's the first one. Let me put that back on my chart. Okay. Now, the second one. What's this picture? All right, so both pictures are now in the chat. Okay, there we go. All right. So again, so what we're going to look at is a monomer, which is the smallest unit of a nucleic acid, and it's made up of 3 parts. It's made up of a nitrogen base, a sugar, and a phosphate. They are linked together. Then you want to take a lot of these nucleotides and you want to link them together. So this one says chains, RNA and DNA are chains of nucleotides all linked together. Okay, so we have two different, so we're going to, when we get to function, we're going to talk about the fact that there are two different kinds of nucleotides. So we've got some things to put in the boxes now. All right, so let's go to Canvas. All right, you want this sentence right here. So actually, let's start with the first sentence. This is going to go in our example column. So you want to copy that. You want to put that over here in our example column. Okay, there's two types of nucleotides, DNA and RNA. All right, then we want the purpose of DNA and RNA is to carry out the genetic code. So that's going to go in our first box. All right, so y'all saw where I got that from. I got that from Canvas and I just copied and pasted it. All right. This second sentence, you can put this one under the picture. So we want to come over here and hopefully I won't make my picture disappear. I did. Ah, okay, I know how to do this though. Put the picture back. What are you doing? I'm having one of those days. Okay, the picture is back. And what I want is to put the words under the picture, but now I got to copy it again. All right, so I want to copy that. I want to put that underneath the picture. There, here. Okay, there you go. Like I said, I'm having one of those days. All right, so a nucleotide is a sugar, a phosphate, and a nitrogen. Okay, which is basically what the picture is showing you. So it really didn't have to do all of that. All right, but words are nice to have. All right. This part you can put in your second column. It has nothing in it right now. So in the second column, you can put down and again, when we do genetics, you're going to have to know this in particular, and we're going to work with these a lot then. And let me see if I can make that a little smaller. Make it 10. There we go. All right. Okay, so there is a difference between DNA and RNA. So note the difference. We got the bases, and then when we say bases, you see where it says over here nitrogenous base. The nitrogenous, so the thing that is the same on all nucleotides is the phosphate and the sugar. They will always be the same. But what will make each one different is the nitrogenous base. So the nitrogenous base can be adenosine on a DNA, it can be adenosine, thymine, cytosine, or guanine. Okay, so each one will have one of those as a base. If it's an RNA molecule, the thing that will make it different from a DNA molecule is that you have two things that are swapped out. They have adenine and they have guanine, but now they have and cytosine, but now it has uracil. Uracil is in place of thymine. So that's going to be what makes them different. That's a test question, folks. They'll give you a picture and ask you, they'll give you the base and ask you, is this RNA or DNA? So you need to know that RNA has uracil and DNA has thymine. All right. Later, when we do genetics, it will be important because when we do translation, when you translate something from DNA to RNA, you're going to have to remember that if DNA has a T, then the RNA is going to have a U. All right. So we'll talk about those later because there's matching pairs and all that sort of thing that we'll talk about. But right now, you just need to know that a nucleic acid is necessary. Its function is genetic code. It's not a food we eat like the other three. It's not a food we eat. It's something that is built in our cells. And it has a nucleotide made up of phosphate, sugar, and a base. And it's one of those four bases. And then when you put a lot of nucleotides together, you form chains of DNA and RNA. Okay? All right, so let's see what else we need to know in the next 10 minutes.
Hi, this is Ms. Delosier, and these are your notes on nucleic acids, so DNA or RNA. So nucleic acids, DNA and RNA, right? We're all familiar with those. That is your hereditary material. It's your hereditary information. It's on your chromosomes. It's in your genes, whatever you want to say about it. It's your nucleic acids, right? And they're made of monomers of nucleotide. RNA is made up of A, G, C, and U, and we're going to review that again in a second. So let's look at what a nucleotide is first. So a nucleotide is made up of a sugar to start. So we have a sugar, and in DNA, our sugar is deoxyribose, which is a 5-carbon sugar. In RNA, our sugar is ribose. So let's look at this 5-carbon sugar, and then we're going to go ahead and we're going to attach it to a phosphate group. Now, because that phosphate is negatively charged, this is going to provide an overall negative charge to my DNA, which means DNA is most definitely charged, which means it is most definitely going to be hydrophilic. So it's going to like water. So I'm going to take my phosphate group, attach it to a sugar, and then it's going to attach to the working end, which is the nitrogen base. And this is a super generic nitrogen base. That's a made up nitrogen base. Don't think there's one that looks like that. So that's my nitrogen base. Okay? And so that, all three of those together make up a nucleotide. When we did these in class, I just had you make the base because we didn't have large enough kits to make the sugar and the phosphate group too. So let's go ahead and let's look at the basic structure of DNA. So DNA, you have a yellow sugar, or you're going to have a yellow phosphate group attached to a white sugar, right? So yellow, white, yellow, white, yellow, white. So basically what I have is I have this backbone going down the sides that's phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then hanging off the sides, hanging off the sides, you end up getting the nitrogen bases. And the nitrogen bases, because DNA is two-stranded, right? The nitrogen bases stick off towards each other and they form like the rungs of a ladder, and they bind together through hydrogen bonding. They are, all of the bonds in what I've got drawn here are covalent bonds, but when I actually have like A and T next to each other, they're going to form hydrogen bonds. Okay, they're not going to actually form a covalent bond across the strands. They form a hydrogen, they actually form many hydrogen bonds. So that's my DNA structure. That's a single-stranded structure, but we made it DNA. It's a single-stranded DNA. So let's talk about the 5 types of nucleotides. So my 5 nitrogen bases, there's only 5 nitrogen bases. There's adenine, which always pairs to thymine in DNA, and then guanine, which always pairs to cytosine in DNA. And then in RNA, I have uracil, okay? So adenine and guanine are what we call purines, which basically just means that they're double ring structures. So see the double ring structures below? Like, don't memorize them. That's a waste of your energy. But they're double ring structures. Adenine and guanine are double rings. Thymine and cytosine and uracil are what we call pyrimidines. And I always remember this because uracil is the shortest of the nucleotides, and it's a single ring. So single ring, but it's the long word. Pyrimidines sounds like it should be that double ring thing, and it's not. So I just remember that I'm wrong all the time, and that the shortest word, uracil, is a single ring and goes with like the long pyramid type thing. That's how I remember it. I don't know how you're going to remember it, but you need to know that. So uracil is going to bind to adenine. So if I have an adenine in my DNA, when I make an RNA copy of that, it would go A to U. And we're going to do activities on that in class. So those are my 5 nitrogen bases. And so in DNA, I'm going to have a double strand, and I'm going to have A, T, G, and C. So I'm going to put like a little yellow check mark next to all 4 of those. And then in RNA is single-stranded, and I'm going to have A, U, G, and C. So I'm going to put a pink check mark next to A, U, G, and C. And so that's your basics on the monomers of nucleic acids. And that's really all I want to talk about on DNA and RNA for these notes, because we're going to spend like an entire unit on DNA and RNA and like how we actually make proteins out of them. I just want you to know the components of DNA and RNA and how they go together. So the backbone is all covalently bound. Across the strands is going to be hydrogen bonding, and then you do need to be familiar with the 5 bases, which ones are purines and which ones are pyrimidines. If you have any questions, come see me in class or send me an email.
All right. She said something that I do want you to add to your chart. So let's go back over to the chart under function. They carry the genetic code, and we're going to add and the instructions. And help it if I spell it right? All right, and the instructions for making proteins, because that's what DNA does. It's gonna be a long process that we talk about, where we copy the DNA and we're going to talk about different types of RNA, and then it goes to the part in the cell called the ribosome, and how the ribosome is a little factory that makes proteins. So the whole point of nucleic acids is to make proteins. We are mostly protein and water. That's what we're mostly made of. All right, so I'm going to stop here. So make sure you add that to your chart, because that is the particular function. So it is the genetic code, and its job is to make proteins. All right, so we'll worry about water on when I see you next Friday. And we'll talk about water, and then we'll be done with, so by Friday, we'll be done with our chart. Okay, we'll be done with macromolecules altogether. So Make sure your chart is complete. It's got everything you need to have on it. Go back to Canvas when you're done with that and do the what you challenge, what you need to know before you go challenge. So know that before you go. Okay. So take a minute to just drag these. Macromolecule involved in the genetic code is no, a nucleic acid. Monomer is a nucleotide. Okay. I should be letting you all tell me. Types of nucleic acids with a double strand made of nucleotides of 4 bases of the master code. Now again, one of the things you do need to know, the DNA is double stranded, and that's what makes it different from RNA, because RNA is single stranded. Okay. And the base that is found in DNA but not RNA is thymine. So that's the list she just gave you. And the base that's found in RNA, it is found in the nucleus of the cell. All right, you need to know that that's a quiz right there. Okay, hint, hint, that's a quiz right there. All right, so there's only a few minutes left to class. Tell me if you need anything from me. I'll make sure you've turned in anything that's missing. Maybe I'll see some of you this evening when you bring your parents to the open house. All right, that being said, I'll see you tomorrow. You all have a great rest of your day. Oops, I'm sorry. You should be on Canvas, not. Hopefully you dragged that properly.